So welcome to the current iteration of the COVID Immunology Consortium Symposium, and delighted to welcome Chris Overall, who's our main speaker for today. So just want to start with a land acknowledgement, say that BC Children's Hospital Research Institute operates on the traditional ancestral and unceded territory of the Coast Salish peoples, including the Musqueam, Squamish, and Tsleil-Waututh nations. And this acknowledgement extends to all Indigenous peoples wherever you are and on whose land we live, work, and play on. I do want to encourage people to fill in the member survey so we can make sure we're continuing to serve the needs of the group. So the aim of the consortium is to bring together researchers and knowledge users at all stages of their career, from trainees all the way through to senior faculty, to help facilitate collaborations for people who are working in the area of infectious disease immunology. So while we started focused on COVID, and still most of the focus is on COVID, I think sort of the long-term goal is to sort of have a broader view on things. And so as part of the consortium, we have collaborators from over 100 researchers across the province. Beth, who organizes most of what we do, puts together quarterly newsletters, and we have these research symposia every couple of months, and we have a very active trainee group, which you'll hear more about a bit later on this morning. So today's presenter is Dr. Chris Overall. So he is a Tier 1 Canada Research Chair in Protease, Proteomics and Systems Biology, and an honorary professor at the Freiburg Institute of Advanced Studies, Albert Ludwigs in Universität in Germany. He was inducted as a fellow into the Royal Society of Canada in 2018, and his publications are highly influential with a very, very impressive H-index of 100. His advances in proteomics have been recognized by the Canadian National Proteomics Network Tony Pawson Award, the Proteomics Scientific Society Award, and the 2018 International HUPO Discovery Award in Proteomic Sciences. He also chairs the HUPO chromosome-centric human proteome project. He's probably best known for his development of proteomic methodology and the discovery of protease substrates in vivo, establishing the field of degradomics, and has used these techniques to reveal new biological processes for proteases in immunity and disease, most recently for COVID-19. And so on that note, I will stop sharing. If I can click the right button here, and I'll let Chris, if you want to share your slides, and then we can get going.
So you can see me there, okay? I can see the slides there, okay?
Yep, I think that's okay. The first slide, yep.
Yep, great. Yeah, so yeah, good morning, and I was just pleased to be invited. I hadn't heard about this group before, so I was saying to Manish before, and it's great to potentially get involved with some other groups here at UBC on COVID. We switched my lab to COVID research 2 years ago, pretty well exactly. I was at a meeting in northern Italy, about 200 kilometers to the east of the epicenter at this exact time 2 years ago. So I was designing the constructs then to express the 3CL protease, and I came back and pivoted my whole lab then to that, enabling us to do the work I'm going to talk about today, as well as 5 other projects, which I won't talk about today. But I want to acknowledge particularly the great help we received from we had great collaborations with Karen Mossman in McMaster, and her postdoc, Aaron J., who's now in Saskatoon. Fabulous postdoc who did the COVID SARS-CoV-2 infections, which we received the lysates from, and set up design experiments with him in the summer last year, while Finder here at UBC was still getting up and running. Also Hong Lin at St. Paul's, and a fabulous postdoc in her lab, Yasia. He was tremendous for helping us with the autophagy assays. And Eric Jan, who many of you will know also, and his postdoc, Marley Volk, who did a few of the transfections at the end with VIRO-E6 cells. We also collaborated with Cecilia Lindskog from the Human Protein Atlas in Stockholm. But the main work was done by two postdocs, a married couple in my lab, Isa Pavlos and Johan Mercado. The lab also, we all were involved in the project, helping out and learning as we went on the way. In very weird times, as you all know, what it was like walking around in empty buildings with only two other labs in operation at the Life Sciences Centre. So what I want to do today is tell you about the insights that we've found into the pathogenesis and evasion of antiviral responses by COVID-2 that we think enable the infection to kick in and to take over the cell. So as you know, there's two ORFs, the frame-shifted polyproteins, there's two viral proteases encoded by COV2. This is 3CL Pro and PL Pro. Today, I'll be talking about 3CL Pro. And together they cleave and release 4 structural proteins, including the famous spike protein, and 16 only non-structural proteins that are necessary for replication. NSP5, that's the 3CL protease. It cuts to 11 quite classic now cleavage sites, but following a glutamine with quite small residues afterwards at P1. As you know, protease from viruses are validated drug targets has been game changers in HIV/AIDS as retroviral protease inhibitors. So these are validated drug targets and great to understand. But I think the deeper question in virology, not being a virologist, you look at this and think, how on earth does 16 NSPs, how do they form the replication complex, as well as doing a whole host of other things, including avoiding immune sensing by the pattern recognition system, avoiding autophagy, blocking interference signaling and ISG responses? How do they escape adaptive immunity, reduce non-essential host transcription and mRNA levels? How do they hijack the protein translation resource of the cell, beat the ER stress unfolded protein pathway, modulate the cell cycle, prevent apoptosis? How do just 16 proteins do this? And it's not just by competition. And to me, being a protease person, it's quite obvious that the viral proteases must be doing a lot more, and not just one or two things more, than targeting the polyprotein, that they're multifunctional and must have a large role in the host cell protein cleavages. The great thing about proteases as compared to kinases, they are irreversible, so the virus can affect irreversible change in the host cell proteome and act at a distance, and from the micron to the meter scale, or tens of meter scales if you're a blue whale or something. So they actually are very effective messengers from a cell outside the body, and within a cell, they can penetrate throughout the whole proteome. And so how do you find substrates for a protease? And the classic way is just going to your freezer and cherry picking favorite candidates and seeing how you go. But this is low coverage and high failure rate. And so some years ago, we applied our technique TAILS, which is a proteomic approach to address this. And we did this with Eric Jan's lab, looking at Coxsackie and poliovirus. We found over 100 substrates for those viruses. So I'm not going to describe the proteomics, just to describe the concept. And so, as you know, proteins have a natural N-terminus, and upon cleavage, you get what we call a neo-N-terminus. And these were techniques that we invented about 10 years ago to do this at scale in complex proteomes and in vivo. We also have a C-terminal approach, which I'm not talking about today, but we've modified and improved, and we're doing this, of course, now with COV2. And this C-tails approach is less effective due to the chemistry, but it's still powerful. So that's it for the proteomics. The way we approached this, as one of the approaches in the lab, we've got other approaches running, was to take initially just cell proteome from HEC293 cells and to digest this with a 3CL protease. Now, the control is the inactive mutant. We just mutated the cysteine to an alanine at the active site. And so this is added to the whole protein lysates. And I want to emphasize that these are native proteins. We know exactly how to do this. We've been doing it for over 20 years. This is a very well-established technique in the lab. And so we did this with 3 complete independent biological replicates. So everything you've seen here, you'll see today, has been reproduced multiple times. And we also did this in human lung epithelial cells for greater relevance. We did this also 3 times. And in addition, we stimulated with type 1 interferons, alpha and beta, and we did that 3 times independently as well. And so I won't describe the data winnowing and so on, but we came up with then a list of 101 substrates, and these are 100% guaranteed to be substrates. I won't go through all the numbers and stuff, but in proteomics, you get thousands of proteins, but we've got a very good substrate winnowing procedure. And we wanted to have high confidence in our atlas of substrates. And what you can see over here is this is found in all 3 of the HEC293 replicates and in all of the replicates here for the B2B epithelial cells. So these cleavage sites were identified by this peptide here after the cleavage site here indicated by the downward arrow. So the peptide, this is the first 4 residues of the peptide. That peptide will extend to variable lengths to the right-hand side here. So they, as you can see here, they were found in 12 out of 12 independent biological experiments. And going down the list, you can see the numbers drop off a little bit. That's just vagrancies of the mass spectrometry. So you might see some of your favorite proteins here that you know and love. And one of them you can see here is TAB1, which is involved in NF-kappa B signaling regulation, and YAP1, which I won't talk about today, but YAP1 is a really important kinase involved in the Hippo pathway. So we validated these by, we generated peptide, we synthesized peptides that span the cleavage site from P4 to P4 prime. And these are, excuse me, these are asymmetric peptides, so they're longer on the right-hand side with the YAYR sequence there. That's on the mass spec. They separate from the cleavage product. And what you can see here, this is the intact peptide here. And then over time, this is at 5 minutes, generates a product, and the product increases with time as the intact substrate decreases. And so this was done in a high throughput multi type of assay, not high, well, it's high throughput, but it's also a lot of work. And from this, we're able to get kinetic parameters, apparent K cat over Kms for 54 of these cleavage sites. So we did this 54 times with the synthetic peptides, and now I've just recently completed this. So all 102 sites and 101 proteins have been validated now by deriving K cat over Km values. So what you can see here, there's a variety of rates. So for instance, here, this is RBM1, 1.5, and so the red here is a cleavage product going up, and I might use the mouse, that's better, the cleavage product coming up as the intact substrate comes down. And so there's rates going very, very quickly, essentially all cleaved in the first few minutes, going down quite slow, but still being cleaved like this MAP4 kinase 5. Well, what's interesting, this is an unbiased screen. We have great confidence in this screen, and we found non-canonical cleavage sites. I mean, a lot of people would just search for the glutamines, and that's all they would look for. But we've found that quite often that proteases have a bit of loose specificity. So we found histidine and methionine even in 10% of the substrates, and these are cleaved with good kinetics. Not as good as some of the best ones up here, but still better than these ones down here. For instance, CREB1, which is a transcription factor involved in the Hippo pathway. So Hugo in the lab, he performed on the ARC supercluster 54 peptide docking simulations for each of these and to see the fit in the active site. So this is the P1 histidine of RBM15 fitting quite nicely in the subsite 1 here. And over here, we've got the methionine beautifully docking in here from MAP4 kinase 5. So this is an example of the data. This is a beautiful spectra here from this very, very long peptide, which is unusual in mass spec, but Tails technique does this quite nicely because the charge, the labeling that we do. So here's a glutamine here in this site here, and you've got the long peptide here, cutting here in PTB1. Now, this protein binds mRNA and is essential for, in other viruses, for viral translation and then replication. And so we found then we were able to obtain this from 5 human primary airways. So these are brushed, so under GA, the human bronchial epithelium is brushed and collected. And from those cells, we're able to show in lysates that the 3CL cleaves very nicely, generating this cleavage product here of PTB1, the products up here as well, but not in the CA mutant, that's the inactive control. So really nice 100% degradation there. Now, in vitro assay, this is recombinant PTB1. You can see here that the intact protein is slowly disappearing over time, going up to 16 hours. And then here, this is cleaved with or incubated with the inactive mutant, the CA mutant, and also in the presence of a 3CL Pro specific inhibitor from John Betteris in Edmonton. And so the PTB is not being cleaved. So pretty good evidence here that we've got nice cleavage. Now in COV-infected cells, it's a VROE6 cells. You can see here very nice PTB fragments being generated and loss of the intact at infectivity of 0.1. There's 3 cleavage products being generated. There's this product here that comes along here, that comes up in time, and these 2 here. So we did Edmund degradation of these. This is the exact, if you remember, the AV, that's the peptide that we found from the by the mass spec, and also this product here, but this is AA, it's a different peptide. And when we look at the map here, it actually turns out that this is the AV peptide. This is in isoform 1, and actually in all 4 isoforms, but isoform 3 of PTB has this insert, and there's a cleavage site here. So the enzyme, the virus, really wants to get rid of PTB1. Also, PTB2 is cleaved. Now, what you also notice here is a nuclear localization sequence. So we've found quite often that processing, this is not degradation, this is cleavage at 1 or 2 sites, very specifically with high kinetic efficiency, and that leaves behind the C-terminal half of the molecule. And it's got a chance now to behave differently, because in this case, we've lost one of the binding modules here and a nuclear localization sequence. So this now, the C-terminal fragment, doesn't have an NLS. So here we have got in the nucleus here of the mock-infected cells, we've got PTB now lighting up quite nicely in the nucleus here. So the rest of the cell is here, right? And now in COP infection, what we have now, I'm showing here is spike protein lighting up very nicely. But one of the problems in doing biochemical analyses or proteomics analyses of COVID is that not all cells get infected. There's and the timing is not synchronized for infection. And so the signals you detect biochemically can be dampened out by the non-infected cells like you see up here. And here, but these form nice controls. So here's PTB in the nucleus in a non-infected cell. And whereas in the infected cells, we've got PTB here coming out into the cytosol. Whoops, I just lost that there. Yeah, so this is, caught actually in the act here of PTB coming out. I just wanted to, sorry, I had a trouble with my slide advance and I still do, not too sure why. Okay, there we go. So here's spike. It's a very strong signal, so we remove that from the channels in order to see. Now you can see the PTB out of the nucleus here in the cytosol. All right, so this is an infection. And down here also, we've got PTB1 caught in the act here of coming out of a disrupted nucleus, all in the cytosol here. Because this is due to the presence or lack of the nuclear localization sequence. So here's the COVID-2 proteins in yellow with 3CL Pro in the center here. What I've mapped here is the 26, or what Johan's mapped is the 26 COVID proteins interacting with 74 of the 3CL substrates. So these are direct interactors where they're touching, the substrates are touching directly these viral proteins. Now this is from databases. So this is from the Groningen Group's paper that came out last year from the interactions. Now this is a direct interaction. And we've also got through with one hop, so through an intermediate protein, a protein complex, the other substrates. So we've got embedded in the proteome now, the substrates, 74 of which are making contact and have been mapped also before by interactomics to be contacting these viral proteins. So I've been talking about PDB. I haven't got time to talk about the inactivation of MAP4 kinase 5, which we showed in the disruption of the Hippo pathway. It's for another talk. I just want to briefly touch upon the galectin story to wrap this up. So galectin-8 is binds glycoproteins. This is the cleavage site we found in 9 out of 9 human lung epithelial cell experiments. This is the modeling that Shugo did. We've got this linker between the two carbohydrate binding domains. There's dropping into the active site, and here's the cleavage site here, fitting really nicely. Now, galectin-8 is the only interferon-stimulated gene product that we found as a substrate. We mapped out those. We found almost full coverage of the interferon-stimulated genes expressed. So this is galectin-8 here in the control cells, quite low amounts. And then upon stimulation with interferon alpha or beta is coming up quite nicely. Now, an infection of Calu-3 cells, we can show here in quadruplicate, the two isoforms of galectin-8 being stimulated compared to the mock infected cells, and with the cleavage products showing here. And this is also showing in 2 of the 5, we did the other 5 as well, human epithelial cells, the nice cleavage products matching up with what we found in infection. So we suspected that galectin-8, because it is involved in autophagy of salmonella, we checked to see if it could bind to galectin-8. This is the binding to galectin-8 by binding to spike protein by galectin-8. Now, when we cleave this and use the cleavage products, this binding drops right down to baseline. And then when we block the binding through the glyco using TDG, a galactoside inhibitor, it also eliminates binding. So galectin-8 binds spike beautifully via the glyco side chains of spike. So it's like a handoff between ACE2 on coming into the cell, then what happens to it, right? It has to get off and start moving around. This is one of the handoff mechanisms, perhaps, but also one of the cell defense mechanisms. So galectin-8 now is recognizing spike. And what it does, it binds NDP52. Now, NDP52, I'm showing here, we showed it binds to the C domain of GAL8 to spike. We assembled this whole trimeric complex. This is it here. This is the trimeric complex here with different concentrations. And upon cleavage, this completely removes the C terminal domain and NDP52. Now, NDP52, this is the autophagy adapter. And what UCIA did, we're showing here now NDP52 in cells. This is the galectin 8 and in the merge. So they co-localize in the cell. So NDP is co-localizing with GAL8. But when we transfect in the cleavage fragment analog, so this is NGAL at the exact cleavage site, or CGAL, there's no, these alone don't bind and light up to NDP52 in the cellular context. And then finally, we were able to obtain from Sweden then some 2 normal lung samples. And so this is the normal lung structure here of H&E. And this is the merge in orange of NDP52 and galectin 8 from a deceased patient at post-mortem. But from 3 COVID patients, now these lungs, they had over a litre each of fluid. It was incredible, completely blown out. And as you can see here, really disastrous looking lungs. And these anti-vaxxers, these truckers, God, just to show them a couple of these sections. Actually, that won't convince them. But Cecilia counted over 5,000 cells to enumerate the statistics for this, and only 5% of these showed co-localization, and mostly there's no co-localization of the galectin 8 or NDP52 in the COVID patient. So what we've got here is that we've got galectin 8 binding spike via the glycochains. This is targeting, I haven't shown this data for viral autophagy and destruction. So then lysosome enzymes come in and it destroys, gobbles up the virus. But the virus evades this by cleaving in the linker of the 2 carbohydrate binding domains of galectin 8. And that breaks apart this trimeric complex. And so now the virus isn't shuttled off for autophagy nor for destruction. So this is an evasion mechanism. And so this balance between the intact and the cleaved galectin 8 may determine severity of infection from severe COVID to mild disease in resistant patients. There's some evidence of SNPs in this site here. So just lastly, I just want to show this, the whole landscape. This is all the substrates mapped in. And we know there's over 2,000 interactors of the substrates, directly or indirectly. And one of the things is that, so there's only 100 substrates. In actual fact, we know that there's 68 candidates we've found here. And most of these we've also shown now are actually substrates. And other approaches in the lab through inactive catalytic domain capture and bioinformatic approaches, we've got probably another 100 substrates. And these papers will be coming out over the next year. But one of the effects, though, is not just disruption of the proteome by selective cleavage of certain proteins, but we've also got these funny interactions here. So what we've got here in magenta are the interactive proteins, in this case between, say, Lima and Caprin-1. These proteins form a complex with Lima-1, those proteins, and Caprin-1, that they're bound together. So you can imagine then that this being cut and this being cut is going to really isolate and change the function of these interactors. We call these stranded proteins because when you've got over, say 300 proteins that are substrates to the enzyme, you're stranding a lot of other proteins that are not cut. And we know several of these, and we've actually investigated these. These are not cleaved by 3CL. We've validated that. So these are stranded and have effects as well. So it's a way that the virus can really send out ripples of effect throughout the whole cell, both directly and indirectly. So look, I think I'll stop at that point there and just leave the summary up. So thank you. I'm happy to take questions.
Thanks, Chris. So while people are just reading through the summary, we can start with the questions, but we'll leave that up as you say. So Kelly, got your hand up, so go for it.
Fantastic talk, Chris. That was great. Hey, probably naive and very general question, but given the sex bias in severe COVID, I was wondering if you ever overlaid kind of sex differential expression of the substrates to see if you can come up with some that actually might give you a clue as to why you see much more severe disease in males. And I was particularly curious about the galectins.
Yeah, I mean, the females in the lab call that male man sickness, right? There's wusses. No, I agree. I mean, sex differences are clearly important, and also from the hormonal response and the cytokine storm point of view. But from this point of view, from here, no clear sex. We didn't look for sex differences at this point. We're just happy to find substrates, but that's clearly something that we'd have to address in the future. What we can say is that there's SNPs involved in some proteins. And what I didn't talk about today is quite a famous protein. We're getting ready to submit this. I'm not going to talk about it now, but this protein is encoded by Neanderthal, part of our Neanderthal genes, and about 30% of us don't have its C-terminal 18 or 20 residues. That isoform is lacking. And there's quite a clear connection between this protein and resistance or not to COVID. And so those with the long isoform are more resistant, for sure, to infection. That's what the studies have shown. We found that there's cleavage of 3CL that removes the C-terminal part. So there's also not just inherent resistance initially, but then later on that can be circumvented by the virus. So there's really genetic differences from SNPs and from mutations and just differences between populations, as well as, for sure, some sex differences, Kelly.
And then another fearful question. So, you know, when you start looking at proteases and always thinking, you know, complement and some of the thrombotic cascades, I know you're being very rigorous in using kind of peptides to validate the targets of this protease rather than something that gets activated by this protease and then leads to cleavage of other proteins as a downstream cascade. But is there a way to kind of dissect that in a big way for all of these substrates?
Yeah, no, that's always an issue in biological systems that you can show, firstly, biochemically, like we've done by recombinant or by peptide analyses, that a protein can be cut. That doesn't mean to say it is cut in vivo. In this case, we were validating our in vivo data, so that was nice linkage there. But in addition, you've got, as you say, and as everyone knows, there's many other proteases, 560 proteases in humans, and any of these can have knock-on effects. We call this the protease web, where one protease can activate another one or inactivate another protease or change the inhibitor profile. And so for sure that can change and cause differences in proteolytic cascades. And this is something that the concept that we've been advancing and thinking about very long and hard for years. In some ways, it doesn't matter if you've got a knock-on effect. Without initiating protease, it wouldn't have happened to begin with. So, but for sure, this is how probably you're getting some of the thrombosis and complement activation that's going crazy. And we have actually a complement substrate also that's in the works, Kelly, that we're wrapping up at the moment in another study. And one of the actually two of the interferons are cut by 3CL. Interferon gamma at the C terminus, but only in a SNP. And that SNP is the isoform that we all use. The isoform that you all buy is actually not, it's only present in like 0.05% of the population from India. Is just by chance, whoever produced that recombinantly, that's what we all purchase. that form is actually cleaved by 3CL Pro. But the form of interferon gamma in us, most of us, by probability is resistant to the 3CL cleavage. But there's another interferon that we're getting ready to publish soon that's inactivated by 3CL.
Thanks, Chris. Yeah, Pauline.
Yeah, Chris, a little bit tangential, but could you tell us a little bit about, like, is it a good target and can it be differentiated from all the other proteases? And what is this Paxlovid that you've mentioned? So sort of how realistic is it to sort of think that this is going to be, you know, it could be a good drug target?
I think anything a virus makes is a drug target by definition almost, because viruses are so efficient with their genomes. Anything they need, they want, is a drug target. But we know that inhibitor in our own hands, when we add that to COVID infection, it stops the infection dead without killing the cell. And this has been done largely in not by us, or we've done it ourselves in our own studies, but also, I mean, this is you know, it's one of Pfizer's Paxlovid. That targets 3CL-Pro and is a validated drug target, and it's safe and effective and reduces I'd like to get with them to show reductions in cleavage of these substrates as a marker for on-target activity of their compound. But I think that there's no doubt that the viral protease is a drug target. I wasn't sure I didn't quite catch the first part you said, pulling from an analog or ortholog, you said, of?
I think it's sort of in general, right? How, I mean, given that there's so many proteases in the cell, right? If you do have an inhibitor, I guess how specific is it to going to be to three?
Yeah. So this is a cysteine protease inhibitor. You have to be careful, but you know, in pharma, basically what we're going for is about 1,000-fold selectivity. So there is always going to be some inhibition of other proteases. Depends on the compound. There's a small Pfizer's a small molecule compound. The active site's nice and attractive for drugging. It's got lots of little pockets and things. So I haven't seen the profile against other cysteine proteases, but the compound we use from Joanne Lemieux in Everton, that's actually a pretty good, pretty selective inhibitor. So the doses that we normally would be using for would have very, very low effects on other cysteine proteases in us. So the calpains, I don't know if they've tested calpains, but that'd be one that you'd want to test. Okay. But in any case, it's better than COVID.
Thanks.
So Chris, I have a question sort of with my pediatrician hat on, in that, you know, I think from the last 2 years, I think most of my colleagues have been surprised and a bit grateful how little this has affected children, and that we haven't seen the pediatric wards filled with kids with severe COVID. And so I was wondering if there was any data relating to the work that you've been doing and the proteomics work that might sort of help explain some of that, and whether there's differences in these pathways in children compared with in adults or in other populations.
Yeah. I mean, honestly, I mean, we were just, we haven't explored that yet. And I mean, there are probably perhaps some isoforms differences, but I think it's probably, I mean, I'm not a virologist, but I suspect that it's more of a difference maybe in the lung structure and the type of, and the ACE2 expression, that there may be differences in kids versus us temporally through development. I suspect it's something along those lines, but you'd have to ask a virologist, Manish, on that, I think, more than a mere biochemist.
Yeah, no, I mean, the ACE2 expression has certainly been hypothesized as one mechanism, right? So, but I was wondering whether there may be other things contributing to that. So it's definitely...
I mean, the interesting thing about ACE2 is that it's not found in lungs, right? So that was actually Cecilia Lindskog, who's our collaborator here. She actually published, that was her paper. There's another group also showing the same thing afterwards, but basically in the lower lung, there's no ACE2. But what we see in the lung, though, is the macrophage is just loaded with spikes. So the Macs probably have ACE2 and are just gobbling it up, and then and being infected, and then driving the cytokine storm through NF-kappa B. But the NF-kappa B pathway is a lot of substrates in the NF-kappa B, and the MALT pathway are chopped by 3-CL. So that's out of control. And so NF-kappa B is running, so you're getting all those cytokines from the MACs and probably from other cells also running quite wildly. So maybe it's probably more the differences that way in the kids somehow than actually the substrates, but more in the cytokine response. But an immunologist is better at answering that.
Awesome, thank you. Any other questions or comments for Chris from anybody? No, some applause from Kelly, and yeah, so I just want to huge thanks sort of on behalf of the group for coming in and sharing your really interesting and incredible work with us today. It's really, really appreciated. So yeah, thank you very much, Chris, and it's been good use of everyone's time, and I'm glad that so originally the plan was to have half of the session for the speaker and then half for the trainee group, but Chris suggested that we give him some more time, and that was definitely the right thing to do, so thank you. But we do have a little bit of time for the trainees, and so on behalf of the trainee group, Bryn McMillan is going to just have a few minutes to talk about what they have been up to. So Bryn, I'll hand it over to you, and I think you have some slides as well.
I do. Let me just share my screen. Great. Can everyone see this?
Yep. Yeah.
Okay.
Looks
wonderful.
good.
Great. Well, hello, everyone, and thank you very much, Dr. Ruff, for that excellent presentation. My name is Bryn McMillan. As Dr. Sadarangani said, I am a PhD student in the Department of Experimental Medicine at the University of British Columbia, and I'm also a member of the COVID-19 Immunology Consortium of British Columbia, and I am excited to introduce to you the CIC BC Trainee Group. We are a group of 5 trainees and 3 PIs and wonderful Beth White as well. And our goal is to bring together trainees at any stage of their graduate training to help facilitate collaborations with individuals who are interested in infectious disease immunology. So we organize monthly trainee engagement events, including journal clubs, work in progress presentations, trivia nights, and more. Each trainee event consists of 2 trainee presentations of any style, like I said, journal club, work in progress presentations, or an informal discussion with time left for questions and or feedback for each presenter. We hope to create an open, a very comfortable and an informal environment where folks can easily share their current and or future work, all the while with the opportunity to present in a more formal settings, if you so choose, at the CIC BC Symposium itself. Our trainee group events will allow for trainees to increase their exposure, to receive feedback from peers and or other PIs, make connections within the research community, and expand their CVs. Incentives will also be provided to each presenter, as well as those who actively participate. These meetings, like many others for the time being, will be hosted on Zoom, and we will send out links for those events about a week before each event. So how do folks participate? So we are accepting abstracts on a rolling basis, in that trainees can submit their abstracts to the link that's shown here on the slide, and choose the date and the presentation style that they would like to present. These abstracts will be reviewed by the trainee committee, and the accepted presenters will be notified approximately 1 week prior to each event. There are deadlines that are associated with each abstract submission, so it's approximately 1.5 weeks prior to the event. So please ensure to get your abstracts in prior to that deadline. When exactly are these events taking place? They will take place from 12 noon until 1 p.m. on every second Wednesday of each month. Our first event is coming up here shortly on March 9th, and we are looking for 2 presenters to showcase their work. So please, any trainees, please get your abstracts in prior to February 21st. And this slide here, this was shared in the most recent CIC BC newsletter, and it has all of the information that folks need in order to find more information on the CIC BC trainee group, to sign up for events, and or who to reach out to ask further questions. We really hope to have a great turnout at these events with trainees from across a number of different research sites, and ask that trainees and PIs alike, please share this infographic with folks who may be interested in participating. I'm happy to send out this this version here if that makes sharing a little bit more accessible. Thank you all very, very much, and we really look forward to seeing you at our events.
Thanks, Bryn. Does anyone have any questions for Bryn? There's a few other people who are part of the trainee group here as well, if there's anything. And it's great to see the energy and the enthusiasm coming out of the trainees, and special thanks not only to all the trainees, but also for Kelly and Pauline for all the work that you're doing with the trainees to keep them moving along. So then the last thing, I just want to remind people, also some people who came late, that we do have a survey going out to all members, and that part of that is for people, for ideas of speakers for the symposia and other things that you'd like to see come out of this group, and all ideas welcome. So I think Beth had previously posted a link to that, but if you're able to just repost that link into the chat, and so people will have that available, and it will go out on the newsletter as well. So please do fill that in. I think it's hugely valuable, and we do read them and pay attention to everything that people say. So that's great. So thanks everybody for your time. Thanks Chris and Bryn for your presentations, and see you all again soon.